Hey guys, welcome back to Now I Know. Today we are talking about difference between phagocytosis and pinocytosis. As we always do, we will compare both of these terms side by side and see what are the differences. So let's begin today's video. First, let's understand the definition of endocytosis, okay? Because phagocytosis and pinocytosis are both the types of endocytosis. Now, endocytosis is a method that cell utilizes to uptake any material from outside of the cell to inside of a cell. Okay, when a cell wants to take some material from outside of a cell to inside of a cell, this process is called as endocytosis. Now basically there are three types of endocytosis: phagocytosis, pinocytosis and receptor mediated endocytosis. Alright, so phagocytosis is a method where the cell takes solid material from outside to inside of a cell. Okay, when the cell takes up the solid material I'm emphasizing because that is a basic difference between phagocytosis and pinocytosis. Okay, in case of pinocytosis, it is the liquid droplets or the dissolved solutes that the cell takes from outside to inside of a cell. Okay, so phagocytosis takes up the solid material, and that is why it is called phagocytosis. Phago means to eat. Okay, and that is why this method is compared to cell eating. Cell eating is phagocytosis, whereas because in case of pinocytosis it is the dissolved solutes, it is called pinocytosis. Pino means to drink. Okay, so it is compared to cell drinking. Just remember this way, like how we need to eat and drink, similarly cell also need to eat and drink, right? Take solid material and also take the dissolved solute. So cell eats and drinks by the method of phagocytosis and pinocytosis. Okay, let's see the next difference. Now let's see how cell takes the material in both the cases. In case of phagocytosis, the cell membrane produces something called a pseudopodia, which we know is false feet, right? It produces pseudopodia to engulf the solid material. Whereas in case of pinocytosis, instead of formation of pseudopodia, there will be invagination. So the liquid droplet or the dissolved solute will be taken up in the cell by the formation of invagination, and again a vesicle will be formed. In that vesicle, the molecules are taken inside the cell. Phagocytosis is a procedure to take up a solid material like bacteria or any other dead cell. So you can imagine that pseudopodia has to be really huge, right? Because when macrophages takes a bacteria is really huge. So the vesicle that is formed in case of phagocytosis is really large. So the vesicle formation in case of phagocytosis is going to be relatively large compared to the pinocytosis process. In case of pinocytosis, it is going to be some dissolved solutes or droplets. Mainly sugar and proteins are taken up by such processes, and this is going to be, the vesicles here are going to be comparatively small. This is one more difference. Vesicles in case of phagocytosis is going to be large. Now we know why, because it takes up really large material. Whereas in case of pinocytosis, vesicle is going to be small compared to phagocytosis. And we also know that when the bacteria is taken inside the cell, like macrophages and dendritic cell, immune cell, what they do, they have lysozyme that digests the bacteria, and by exocytosis they are going to throw out the waste particles. So phagocytosis is seen mainly in immune cells like macrophages, dendritic cells, neutrophils, basically the cells that carry out the process of phagocytosis, and thus they are also called phagocytic cell, and they take up bacteria or dead cells. In case of pinocytosis, which is mainly seen in case of liver cells, kidney cells and epithelial cells. So sugar and proteins are the molecules that can be taken up by the process of pinocytosis. So there you go. These are the main differences between phagocytosis and pinocytosis. Phagocytosis is cell eating and pinocytosis is cell drinking. Cell eating because it takes up large solid materials, and cell drinking because it takes up liquid droplets or dissolved solids from outside of a cell to inside of a cell. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I will see you next time. Until then, keep learning.